students, myself Dr. J. Rani, Associate Professor of Zoology, V.V. Vanneperumal College for Women, Virudhunagar. Today I am going to deal about multiple alleles, which is a very important topic in genetics. First of all, we must know what is the definition for multiple alleles. It is nothing but it is a set of 3 or more genes controlling the same character but located on the homologous chromosomes. You can see the diagram in this slide, which is having a same shape, length, and the location of centromere also same. So what is the definition for homologous chromosomes? It is also a pair of chromosomes have the same shape, same gene sequence, same location and same centromere location, and the length is also same. Simply we can say that the 2 chromosomes in a pair, normally one is inherited from mother and other one is inherited from father. So what are the examples of multiple alleles? So here we can see the examples. First one, ABO blood group in man, RH blood group, nature of wings in Drosophila, coat color in rabbit, self-sterility in tobacco. Today I am going to deal with the ABO blood group in man. So what is the blood group? So first of all we must see who found the blood group. The famous scientist Carl Landsteiner, a Austrian-born American biologist who found the blood group in man. So he is called as father of blood group. He discovered human ABO blood group according to the presence of antigen on the RBC. RBC, you all know that it is a red blood corpuscles. He found that substances in the blood, antigen and antibodies, are responsible for the clumping of red cells, that is known as agglutination. So when red cells of one type is added to another type of red cells, there will be a agglutination. It leads to red cell agglutination. Now we can see what are the types of blood group. We all know that there are 4 types of blood group: A, B, AB and O. So A type is the persons who is having antigen A on RBC. They are all known as A group persons. The persons who are having B antigen on their RBC, they will be B group. If they have both antigen A and antigen B, the persons will be blood type of AB. O group persons, they have no antigen on their RBC. Now we can see the tabular column which shows the distribution of antigen antibody in ABO blood groups. In the first column we can see the group A, B, A, B, O. The next column antigen which is present in that group, and the third column we can see the antibody. The person who is having A group, they have A antigen but B antibody. The persons who are all having B group blood type, they have B antigen but A antibody. The AB blood group persons, they have both the antigens A and B but no antibody. The O group persons, they have no antigens but A and B antibodies are present. So according to the presence of A antigen and B antigen, the group is divided into 4 different types. So what is responsible for the typing of ABO? How we can classify? So there are 3 alleles. They are responsible for human blood groups, that is LA, LB and LO. So L denotes the scientist, the Landsteiner, who found the blood group. We can know that the antigen A, it cannot coexist with antibody A. Same thing, antigen B, it cannot coexist with antibody B. So the synthesis of antigen A is controlled by a dominant allele LA, and synthesis of B antigen is controlled by dominant allele LB. And the absence of antigens, it is due to the presence of a recessive allele LO. So these 3 alleles LA, LB and LO, these are responsible for ABO blood group. So here out of 3 alleles LA, LB and LO, LA and LB are dominant alleles and they are codominant. What do you mean by codominant? Codominant means if these 2 alleles are present, I mean LA and LB are present in the same person, both antigens are expressed. Both antigens will be produced by that gene. So that is why this LA and LB are known as codominant. But LO is the recessive allele. If LA and LB are present in the same man, that person is having blood group AB. LA, LB and LO are located in the same locus. That means in the same place. So they are called multiple alleles. In the first hour itself, we can say that the multiple alleles are the 2 or more genes which are present in the same locus we have studied. So here LA, LB and LO both are present in the same place. So they are known as multiple alleles. So here we can see the genotype of A group, B group, AB group and O group. A group person have the genotype of LA, LA or LA, LO. B group persons they can have LB, LB or LB, LO. AB group person have only LA, LB only. O group persons have only LO, LO. So here the ABO blood groups they follow Mendelian inheritance also. Here we can see the tabular column which shows the father, if the father is A group, if mother is also A group, what will be the blood group of child. So this table shows all the types. If father is having A group, B group, AB group or O group and mother is having A, B, AB, O, we can easily find out the blood type of child. What are the applications? Why we are studying ABO blood group? So first of all, we all know that blood transfusion which is very important during emergency time. That is during accidents, during major surgery, for anemic persons. So that for that time also we need blood transfusion. What do you mean by blood transfusion? Means transfer of blood to the patients. So here the blood transfusion is a life-saving process. The persons who are all met with accidents, major surgery and anemic persons. The donor and recipient. So donor is who donates blood, recipient is who receives blood. So second application or second usage, disputed parentage. So disputed parentage means if any child is claimed by 2 parents, we can easily identify who is the parent of that child by testing their blood group of these 2 parents. We can solve the problem easily. So this is known as disputed parentage, which will be easily solved by taking their blood type. Then identification of culprits. In the forensic medicine, in the place of murder, they will collect the blood stain and they will find out the blood type of that culprit and easily they can solve the cases. And here we can see the who is universal donor and who is universal recipient. We can say that blood group O is universal donor because this blood group O, they don't have A or B antigens in its blood. So because antigens only evoke a allergic process, okay. So because in O group there is no antigens, we can donate. The O group persons, they can donate blood to anybody. So O group is known as universal donor. AB blood groups, which is a universal recipient, because they have no antibodies A, B or nothing. So it can receive any blood from anybody. And we can see what are the rarest blood group in the world. AB negative, AB positive and B negative. These 3 groups are rarest blood group in the world. Next we can see the RH factor. This was also found by Landsteiner and another scientist Wiener. It is also controlled by multiple alleles. It was discovered in a rhesus monkey. So the name RH factor. It is also have no natural antibody, but RH negative persons, they will develop RH antibody if they receive RH positive blood. So what are the application and what are the uses? Why we are studying RH factor? First of all, we must know the disease erythroblastis fetalis. It is a hemolytic disease of newborn baby. So here, why the newborn baby is having this disease? Because if RH negative girl marry RH positive man, their child will be RH positive only, but that the child is having they will be suffered by erythroblastis fetalis, that is suffered by jaundice and anemia due to hemolysis. Hemolysis means destruction of blood cells. The baby will be the RH antigen will enter the mother who is RH negative, because the fetus is RH positive but mother is RH negative. But during development, the RH positive fetus, the antigens of the fetus will enter the mother who is negative. So the negative mother, they will develop antibodies against RH plus antigen. Then again the produced RH negative antibodies will enter the fetus and it will destroy the RBC of fetus. This leads to erythroblastis fetalis. So because there is a destruction of blood cells in fetus, the liver will receive damaged RBC, so it is doing so much work. So the liver is heavily damaged and it leads to jaundice, and again it leads to anemia. So how we can prevent erythroblastis fetalis? First of all, the RH negative lady should marry RH positive person. Next, second one, by injection, by applying injection to the fetus. Destruction of RH positive fetal cells in the maternal blood can be controlled by anti-RH immune globulin injection, which prevents RH incompatibility. Again, we can treatment. The treatment is nothing but exchange of intrauterine fetal blood transfusion. The blood is transferred from the fetus. If severe fetal anemia is detected, the delivery can be done at 32 to 35 weeks depending on the clinical situation. So I can conclude my session. Why blood group? Blood group is an important phenomenon in day-to-day life. So we should know what is our blood group. So only we can safely donate our blood or receive blood from others during emergency. So knowledge about blood grouping is vital for all the human beings. Thank you.